This is Module 3, Oxygen Binding Proteins and Methods for Analyzing Proteins. In this video, Protein Sequencing, I'll be going through the different steps and stages of sequencing long protein strings based on their amino acid order. And I'll describe two different ways that we use, or two different methods for sequencing the order of amino acids in a protein. Okay, so in general, all protein sequencing experiments involve three different stages: a fragmentation stage, a sequence determination stage, and reconstruction. So when sequencing a polypeptide, the first thing you have to do is denature it into its primary structure. And so the first thing you have to do is reduce the disulfide bonds to separate any protein that might have a quaternary structure. So you were separating the two chains from each other. It also helps to denature a protein from its tertiary structure into secondary ones by breaking those disulfide bonds. The next step would be to use chemicals or enzymes to break up each of these chains into smaller peptides, and then you would determine the sequence of each of the peptide fragments. Once you determine the sequence of each of the individual peptides, you then try to line up all of you line them up by areas of overlapping sequence. And so this one right here is this fragment here. This one is this fragment. And this one and this one both overlap a little bit with this longer fragment, which is taken from the second pool. So it's actually a very good idea that when you're fragmenting your polypeptides to use two different methods in order to generate different sets of overlapping fragments, so that when you try to stitch them back together to determine what the overall order of amino acids is, you have good reliable overlap allowing you to stitch them back together in the correct order. So this is the general approach to how you would do a protein sequencing reaction. But there's some setup stages that have to happen first before you can start going through this process. The first thing you have to do is determine the percentage of each amino acid in a protein. And so this is a little bit separate from what we're doing here. So we have to first break up the protein in its native state, so in its folded state. You still have to do that, break the disulfide bonds and separate the chains. And then once you separate the chains, before you progress onto this next step in the workflow, it's a really good idea to determine the percentage of amino acids for each polypeptide. So how much glycine is in here, how much tyrosine is in here, how much histidine is in here. And that will help in the very last stage of the process, which is reconstruction. So the first thing we have to do is, before we start to break up those separate chains into smaller peptides, we have to determine the percentage of each amino acid in each protein chain, in each polypeptide. And the way we do this is by using a chromatography approach. So first, you start with your long string, your long polypeptide, and then you break it into individual amino acids by breaking up all of the peptide bonds that hold it together. This is referred to as polypeptide hydrolysis, and you can use acids to break those peptide bonds, you can use bases, or you can use enzymes that specifically bind to and break peptide bonds. And so now what you're left with is a pool of individual amino acids. Next, you can use ion exchange chromatography to separate out each amino acid based on its charge or its isoelectric point by manipulating pH. And so review how ion exchange chromatography works by watching video 3-1, which talks about commonly used lab methods. And one of them is ion exchange chromatography. Just to remind you, if we're using, say, anion exchange chromatography, any positively charged protein is going to be attracted to a negatively charged bead in this column and get stuck up in the column. And then negatively charged proteins or neutral proteins will flow through. And so instead of discarding the flow through this time, you're going to want to hang on to that flow through, which represents all of the negatively charged and neutral amino acids in your system. Then you would elute out all of the positively charged ones and then continue using different types of chromatography to separate out the different positive charged amino acids. Then you can come back to your first waste product, which contains your negatively charged ones and your neutral ones, and then put them through cation exchange chromatography, where the negative charges are going to get caught up on negatively charged protons are going to be attracted to positively charged beads in the column. And so it's a stepwise process. You have to use multiple different types of chromatography based on pH changes or based on charge in order to determine and end up with 20 different pools of distinct amino acids. Now, once these pools are separated and you have your 20 different test tubes, each with 20 different amino acid pools, you can then use fluorescing reagents, add them to your pools, and that will help you determine the quantity of each amino acid in those pools based on how much fluorescence they produce. And so now at the end of this, you can determine the percent of each amino acid in your overall polypeptide chain. Okay, now backtrack to the original workflow. So we're backtracking to this point. So now we've got 2 polypeptide chains for this particular protein. We've just determined the percent of amino acids, each amino acids in this one and in this one. But we destroyed some of that sample by doing that, right? We broke it up into individual amino acids. So now we're going to go back to our sample where we have this long polypeptide chain. And then we begin the fragmentation stage. This means we have to fragment that protein into smaller peptides by using specific enzymes like trypsin. Trypsin is one enzyme that is highly specific for positively charged residues. And so it will bind to those and break the peptide bond between that positively charged residue and its neighbor. There's other enzymes that break peptide bonds based on different residues. So this one, chymotrypsin, binds to bulky hydrophobic residues like phenylalanine, tryptophan, and tyrosine. Elastase binds to small neutral residues like alanine, glycine, serine, and valine, and so on. And so you can fragment your polypeptide into smaller peptide chains. And then again, like I mentioned before in the previous workflow, you want to do 2 different methods of fragmentation so that you get good amounts of overlap in the products. Typically, what you're looking for are peptide chains that are about 50 amino acids long before you progress to the next stage. Okay. Now, on the next stage, you want to separate and purify the fragments using a special method called 2D gel electrophoresis. 2D gel electrophoresis separates proteins, or in this case, your short peptide chains of 50 amino acids, based on 2 biochemical properties. One is that it does it based on its isoelectric point of the overall 50 amino acid long peptide. And then the second stage separates them based on size or mass size of the 50 amino acid peptide. So the first dimension, you're basically passing your peptides through a gel. And within the gel, we have a range of pHs where at the beginning of this process, the pH is very basic. And then gradually, as the peptides move through this gradient, it's moving through a more and more acidic gradient. And eventually, the protein will stop moving once it reaches its isoelectric point, or the pH at which the isoelectric point is reached for that peptide. What this means is when a peptide is at its isoelectric point, it no longer has a charge, right? Because that's what isoelectric point is. It has no charge. And so once the peptide reaches that point of no charge, it stops migrating through the gel. And so now you know what its isoelectric point is of the peptide. Then you essentially turn it and then neutralize all the pHs to a single neutral pH, add SDS to all of these proteins that have been focused by isoelectric point, and then separate them purely by mass. So adding SDS to all of these is going to give everything a negative charge. And so you first separate based on charge and isoelectric point, then you separate based on mass. So here we've got a protein sample that goes into the gel. This one was done in the opposite direction. So it's first separated by mass. And then SDS is removed from that. And then it's separated by, isoelectric point. I don't know why somebody would do that. That's actually much more difficult. So what I'm going to do is just flip-flop. Oh, it's an image, so I can't flip-flop it. basically what I want you to understand is that this does actually need an order. You can't do one before the other. So you have to first separate based on isoelectric point, then give everything a negative charge and separate based on mass. So the order of these two stages is reversed. So reverse them in your notes. So again, you first separate based on isoelectric point, which is going to push proteins in this direction through this matrix. And then you're going to apply an electrical current in the perpendicular direction and separate proteins based on mass. Large proteins will be stuck up at the top, small proteins will be stuck or will flow through to the bottom. And then this is actually what it looks like, where it represents different spots on a gel, represent different proteins of distinct isoelectric points and mass. Okay. So you fragmented your polypeptide into fragments, and now you're separating your fragments based on charge and mass. And so the next stage is to determine the order of amino acids in each of those peptides. And so you would take one of these spots that represents one peptide and subject it to what's called Edmond degradation. There are actually two ways to sequence a fragment. One is Edmond degradation, which I'm going to describe now, and it's the older method, the more traditional method. And then another way is through mass spectrometry, which I'm going to talk about after this. Okay. Edmond degradation involves taking your peptide. In this example, we have 5 amino acids in this one peptide. In reality, it would be 50 amino acids. You then label it with a reagent called Edmond's reagent. That attaches to one end of the peptide, the N-terminus actually, specifically. And then you release the N-terminal amino acid by cleaving the peptide bond that connects it to its neighbor. And so the first round of Edmond degradation will cleave off the labeled peptide. Then you would add more of the label of the Edmond's reagent, do another cleavage step where you break that off, and then so on. And each round, between each of these rounds of labeling and release, you sequence that single amino acid using chromatography methods. Each amino acid has its own signature chromatography pattern. And so you can determine, based on what previous people have done and determine what the patterns are, what the identity of your amino acid is when it's attached to this Edmond's reagent by subjecting it to chromatography. So you would do that after each round. And then you can determine what, you determine the order is starting with the N-terminus and working your way through to the C-terminus, one at a time. Mass spectrometry is another way that you can sequence proteins. And so you first start with separating your protein into short peptides. So we already did this in preparation for Edmond degradation too. Then you subject each of these peptides to what's called ionization. So each of these peptides are ionized, and then they're pushed through an electrical field. And their migration through the electrical field depends on its mass and its charge. So the identity of the peptides are determined based on specific mass to charge ratios. So once we determine what the mass to charge ratio is for each peptide, you get this profile at the end of all of your peptides. And then computer alignment programs will match the profile that you get based on mass to charge ratio to known profiles submitted to databases online. And so you would match the profile to those databases online, and you would learn what your peptide order is based on that. Once you determine the sequence of your peptides, you would have to reconstruct them. And so that's to some extent what this program is already doing for you. It sort of reconstructs and identifies the protein based on the mass to charge ratio of each of these peptides. So mass spectrometry does all of this. This is the entire workflow for sequencing a peptide for using mass spectrometry. But after Edmond degradation, you're basically sequencing each peptide individually. And so what happens next is you have to align them. And so what I'm talking about now is this stage where you determine the regions of overlapping sequence. So you don't necessarily have to do this reconstruction step with mass spectrometry. The program is doing it for you. But if you used Edman degradation, you end up with peptides whose sequence is now known, generated by the first cleavage method, peptides whose sequences are now known, generated by the second cleavage method. And so now you would take all of the blue peptides and try to overlap them and determine what the overall sequence is based on overlapping regions. And that is it. And so just again, to remind you that the mass spectrometry method is determining the mass to charge ratio of these peptides and then lining up the profile for each of those with matching profiles online. For Edman degradation, you have to determine what the sequences are for each peptides in these two different pools and then line them up to determine their overall sequence. And then you can take this sequence and match it to known sequences online to determine what the nature of that protein is. So is it a polymerase? Is it a, other types of proteins?